Okay. Well, let's go and get going. That's right. It's that time. So some announcements. I think some significant ones. So hopefully you all saw the announcement on Canvas. I've decided to go ahead and delay, first of all, Quiz 9, which was going to be due tonight. I'm going to delay that until Friday, so that way I know that we're going to be through the material for that quiz. I've also decided to delay Exam 1. So the reason being that, again, we had the snow day, so we're going to move Exam 1 back from the 15th to the 17th. The other reason is that CERC is going on that day, so a lot of you are going to be presenting your research in that, and I didn't want to pull you away from that for an exam. Okay, so and I didn't realize that CERC was going on that day. Okay, so we'll have that on the 17th. I'll continue to remind you of that, but we're going to have that on the 17th. Okay, I posted a problem set as well yesterday. That's going to be due on the 10th, so a week from today. Okay, and we'll definitely have covered the material for that probably by the end of the day, to be honest with you, or close to the end of the day. Okay, and then finally, recitation sessions. The two tomorrow are going to be held in AZE 210. Okay, so I moved that room. It's going to be in that room for the rest of the semester. Okay, the one on Sunday is still going to be held in the MRB building. Okay, okay. So, all right, our riddle pronounced as one letter and written with three. Two letters there are, and two only in me. I'm double, I'm single, I'm brown, black, blue, and green. I'm red from both ends and the same either way. What am I? I. That's exactly right. An I. Very good. All right. I didn't say they would get any better throughout the semester. I just said that they would be here. Okay, so anyway. All right. So on Monday, we stopped right here, and we had finished discussing essentially the overview of oxidative phosphorylation, looking at the electron transport chain and the ATP synthase and some of the thermodynamics in regards to that, as well as the fact that those two processes are coupled. We then jumped into looking at each of these electron carriers or redox centers that we will encounter in each of the complexes in the electron transport chain. So our job today is to walk through the mechanisms that will allow electrons to move through these complexes, ultimately ending up on oxygen making water, okay, which would be the end step in cellular respiration. All right, we're not going to go in gory detail on that, but we're certainly going to go in enough detail to give you some appreciation for how electrons can move through this system. Right? In terms of protons and proton translocation across the membrane, we're not going to really look at mechanisms for that because, number one, they're not very well understood, and if they are, they're fairly complex. Okay, so and not very fruitful to go through in a class like this. Okay, so so we'll just kind of say that they move across, and we'll go from there. Okay, all right. Now let's go ahead and start here. I've already asked that question, and we'll look at then, of course, complex 1. And each of the complexes, I'll have a slide that will start something like this. Then maybe some supporting slides after that. But complex 1 is where, of course, we're going to start. The complex 1 also goes by the name NADH dehydrogenase. So that's a name that you would find specifically in primary literature. And the reaction that it catalyzes, and this will be true of any of these slides that I start off with the complexes, the total reaction is this. Okay, it's going to take NADH, it's going to oxidize it. Okay, so that would be oxidation, of course, removing the electrons. And it's going to pass those electrons onto Q, or ubiquinone, okay, making ubiquinol, and this will be reduced. Okay. As we look at this, there's a total of 6 protons that we have to keep track of. There's this proton here, and then there are these protons here. You'll notice that I put this subscript in next to that proton to indicate that those are protons that are originally found in the inside, okay, or the matrix side of the mitochondria. Those 6 protons, 4 of them are going to end up directly on the P side. We would say that they are pumped. And that would be the term that's most commonly used to say that they have been moved from inside to P side. They've been pumped. Okay. And then the other two, so we've got 6 over there, the other 2 are going to end up sitting on ubiquinol, really, okay, or the coenzyme Q molecule. Okay, we'll see the fate of those when we get to complex 3. Okay. But in totality, that is our first complex. Okay. Now, there's a lot of steps between these two, or between those, in those arrows, and we're going to look at that, of course, as we move on. Okay. Now, the complex, just structurally speaking, okay, in mammalian systems is made of 45 subunits. So big, really big. Okay, we do not understand the function of all of the subunits, okay, but there's 45 different subunits in it. Within those 45 subunits, there are 9 iron sulfur centers. There is 1 FMN molecule. Okay, and of course, those are things that we discussed on Monday. There is then a binding site for NADH, and there's a binding site for ubiquinone. Okay. Now, I'm going to go ahead and diagram, and we're going to spend probably our most time diagramming complex 1, okay, to give you some idea of mechanistically what's going on here. And then we won't spend as much time certainly on complex 2. Complex 3 is different, so I'll show you that when we get there. But let's go ahead and draw, or I'm going to go ahead and draw here a high resolution structure of complex 1. Very high resolution. Looks like a gun, right, or an L, or something like that, okay? It actually, structurally speaking, and I'll show you, I will show you the high resolution structure on the next slide of this, okay? But that is essentially what it looks like if you were to draw a very, very rough outline for this, okay? I'm going to put the P side on this, and I'm going to put the N side here. Of course, in between here, these would be the 2 leaflets of the membrane, okay? So it has a fairly significant membrane spanning domain. Okay, and then it has also a very significant domain that really is found on the inside or the matrix of the mitochondria. Okay. Now, in terms of the reaction, let me go ahead and do this. This is, we'll try and do our best to illustrate this. I actually have a video of this that will do a better job than what I'm going to show you. But I'm going to go ahead and draw it out anyway, and then we'll look at the video after this, okay? But when you look at this here, we'll start at NADH, of course, and a proton, okay? And it's going to give its electrons up, and NAD plus will be generated, okay? Now, it will give its electrons to FMN. So FMN, if you remember, from our discussion on Monday, can accept 2 electrons as well as 2 protons. Okay. The chemical details of how that occurs is not something that we're going to look at. Okay. But we'll just say that that happens. Now, this next step, okay, is going to be the oxidation of FMNH2. So we have the reduced form of the flavonucleotide, and that has to then give its electrons into really a chain, a fairly significant chain, long chain of iron sulfur centers. Okay, and so what we're going to see here, I'm going to go ahead and do this first. Right? It will oxidize in 2 steps. So it will go from fully reduced to the radical form. And in doing so, it'll pass its electrons, okay, onto an iron sulfur center. Okay, so I'm just going to put it as iron, and let me use a different color to get a little bit more contrast here. Let's do this. Okay. It's going to pass it to iron 3+, making iron 2+. And that would, of course, be a reductive reaction. I'm not putting the S here, but this is an iron sulfur center. Okay, I'm just representing the iron. Now that iron sulfur center that's in immediate proximity to the flavonucleotide is one of the 9 iron sulfur centers that we find in this complex. Okay, there are obviously 8 more. Okay, and they will start, the electron that was just passed to this first iron sulfur center will start to jump up that chain of iron sulfur centers. Okay, so what would be immediately after this or following this is that this iron, okay, would be oxidized by the next iron sulfur center in the chain that will then pick up the electron. Okay? And electrons will jump from there. Okay? Now that leaves this radical form here that still has to be dealt with. That radical form, okay, is going to give its electron again, okay, to the iron sulfur center, but it can't do it until that iron sulfur center releases the electron that it originally got. Okay, so it has to happen in a stepwise fashion. And it's kind of difficult to demonstrate here. I'll show you that video. Okay, but these electrons separate then, and as they separate, then they'll start to hop one at a time up the chain. Now, this is one of the reasons that I spent some time on Monday saying this can adopt the radical form. Okay? And the reason being that NADH can't. It doesn't adopt the radical form. When it gives up electrons, it gives them 2 at a time to flavin mononucleotide. And then flavin mononucleotide has the capacity to split those electrons and give them 1 at a time to this chain of iron sulfur centers. Okay. So these flavonucleotides, and this would be true also in complex 2, it'd be FAD there though. They act as transformers between 2 electron donors and 1 electron acceptor, okay? Because they can adopt these radical forms. Now coming back up to this here, this of course is going to be oxidized, and we're going to say this. We're going to say this happens about 6 times more. Okay. I think that's right. Well, no, 7 times more. Sorry. Okay. 8 of the 9. Let me say this. I think that I did my counting right. 8 of the 9 iron sulfur centers are going to be used in electron transfer. 1 of them isn't. Yes, question.
What makes it energetically favorable for the electron to jump from center to center for it versus just staying at that first one?
Yeah, okay, so the question is why? Why does it do this, right? And the reason is because, let me go ahead and draw this next step, okay? This next step, we have ubiquinone, okay? And it will pick up one of these electrons. Oops, I should have drawn the arrow the other way, sorry. I got that whole thing backwards. The arrow's going this way. Okay, and it will pick up this proton. So going back to your question, that, okay, is very exergonic. Okay, so if you imagine, and I'm not entirely sure of the reduction potential of each of the iron sulfur centers, even if they're the same. Let's say they are the same, and they're probably slightly different. All right, the reduction potential of ubiquinone is quite a bit larger than the reduction potential of the last iron sulfur center. And so it starts to then pull electrons from that. So even if this whole process is in equilibrium, because it's being drawn in that direction, right? According to Le Chatelier's principle, the electrons will continue to be sucked that way. Now it's possible, and I don't think this is the case, at least from some more of the recent readings I've had on this, that these are different in terms of reduction potential as well. Okay, from what I understand, that these are pretty similar in this middle there, and it's that last step that's really exergonic. Now, that's, right, if you look at this here, we have the iron being oxidized, this last step in the chain being oxidized, giving up its electron to ubiquinone, but it only makes a semiquinone radical. So that has to happen again, okay, meaning this, and this is where my drawing is going to be really not very good. So this is going to do this again. It's going to pick up another electron from the chain and become QH2, and there will be another proton that is needed for that. Okay. This QH2 will then go on to complex 3. Okay. And you'll notice here that there has been now 2 protons that have been put on ubiquinone, making ubiquinol, and those are now being carried through the membrane to complex 3. We'll look at the fate of those at complex 3. Okay? Now, at the same time, going along with this answer to this question that was asked, because this process that I just depicted at the very end of this, because that is such an exergonic process, meaning the reduction of ubiquinone from that last iron sulfur center, that can actually drive conformational changes in the protein. Okay? And the current prevailing hypothesis is that there is this long, I mean, structurally we know this is the case, there's this long alpha helix that extends essentially the entire distance of that transmembrane portion of complex 1. That exergonic reaction drives conformational changes in this to go in this direction, and it comes back like this, and it's been likened to in the literature as like a piston in a train engine, right? An old train engine is going back and forth, back and forth. And as it goes back and forth, it allows conformational changes that will essentially allow for protons in different locations to move through this membrane, and there'll be 4 of them in totality. Okay? And they'll go right through the protein. The chemical details of the model that I'm referring to are not nice. Okay? You have to do a lot of structural analysis of the protein to even begin talking about the chemical details of them. I'm not going to do that. I have a slide on it. I'll go ahead and skip over it, but you can look at the paper and, if you want, on your own time. Okay? Questions on this? Yeah?
So what exactly allows the protons to move through protein? What part exactly?
Well, okay, so the question is, what allows the protons to move through the membrane? The first step in it is this exergonic reduction of ubiquinone, or coenzyme Q. That drives, according to the model, drives this conformational change in this helix to go back and forth. There are subunits. What you're not really seeing here is there are subunits at each of these arrows. And the movement of this helix is then actually changing conformation of those subunits. And protons, okay, go through these, or actually amino acid side chains in each of those subunits go through these successive acid-base chemistry reactions until finally a proton sitting at the surface of the membrane, of the outer leaflet of the inner membrane. Okay. And so we're skipping a lot of details. We're just drawing it as blue arrows. So, okay. Yeah. Other questions? That model was just recently proposed. We didn't have structural information on that transmembrane portion until about 2010. So somewhere, it's a recent model. Now, let me go ahead and go here, and then we'll look at a video of this. Okay, so this is it's really the structure of complex 1, but from a prokaryotic organism. There's only about 14 subunits here versus 45. We don't have structural information on the full 45 subunits of the mammalian complex 1. But we do of a bacterial organism, which has 14 subunits that are homologous to what we find in mammalian systems. Okay. What you see here, let me go ahead and do this. These here put a circle around all these. These are the iron sulfur centers, okay, that I referenced on my last slide. This here is FMN, okay. And then what's not shown here would be essentially, this would be where Q binds. Okay. So electrons then really, if you were to follow along with what I did, electrons will come here. They'll start as NADH plus a proton. Okay. Give those electrons FMN, and then they'll kind of shift their way up through this whole process. Okay. Until finally they get to here. At the same time, these protons then that I was referring to, these are going to move through, the way I drew this, it would move through that, through that, through that, and through that. Okay. This here is this helix that I was referring to. Okay. Now, I'm going to go ahead and I'm not going to talk about this, okay? If you're really interested in this, this is a figure from the paper that describes that model. I unfortunately didn't put the citation. If you want it, let me know and I'll get it to you. Okay. I'm going to skip this, but this is the model that they talk about in that. Okay. All right. Now let me go ahead and show you a video of all this. That's not the video I want. This is the video I want. Okay. So this actually is a really cool video. It's older though. In fact, some of the information on this is not quite right. The full structure of complex 1 when they made this video was not understood. So they are lacking essentially that entire portion that I was just talking about, the transmembrane portion. Okay. But what you're looking at here is, of course, oxidative phosphorylation. We have over here in this nice color here, that will be complex 5 or ATP synthase, and then 4, 3, 2, and 1. Okay, we're starting the video about 3 minutes in, so there's quite a bit of information before this. But I want to show you this, at least for complex 1, and then we'll move on, okay? So let me go ahead and do this. By the way, yellow little spheres down here are protons. Okay, so this is the P side. Okay, this is the inside, which is it's flipped compared to the orientation I was showing you, unfortunately. Okay, so protons would be pumped in this direction. You'll learn very quickly that electrons are blue spheres. Okay, let's go on. Oh, I don't want his voice on here or their voice on here. Okay, just a second. I've muted it here, but it's like persistent. So let's do this. Audio, where's audio? Way down. All right, there we go. So let's go ahead and do this. So it's going to focus in on complex 1 first. So here we go. Oh, it's jumpy. Awesome. That's okay. We can survive with jumpy. All right, and let me pause it right there. Stop. Okay, great. All right, so what you're looking at here, that top molecule with the 2 blue spheres, that's NADH. The 2 blue spheres are electrons. Immediately below that, you can kind of see the chemical structure, that's FMN. Next to this, all these other purple guys, okay, that some of them have these blue spheres in it, those are the iron sulfur centers. Okay, so what you'll see here when I push play, you'll see the electrons jump 2 at a time to FMN, and then they'll start jumping 1 at a time to the iron sulfur center. Okay, and you have to say ooh and ah, ooh, and then it goes, and then ah, and then there he goes, right? And it just keeps doing this. This is a lot of fun, and we can just watch NADH keep giving electrons, and they just jump down this chain. That's what I was trying to show with my arrows. It's not as good as this. Oh, they even tell you redox centers. Awesome. Okay. All right, so that's what's going to happen. Now they're going to go on and talk about other things here. I'm going to go ahead and skip some of that, and I'm going to move right to this. Where is it? Yeah, there we go. Okay, so we'll press play here. They try and show how protons are moved. They show this like green magical ether that moves protons through. It's really magical. I mean, biochemistry is magical for a number of reasons. not the least of which is some of our descriptions of these things. All right, so what we're going to do here is we're going to focus in on what's happening down here, okay, and we're going to go here and pause it. Okay, so that thing that you just see here, I'll outline it with my hand, this is coenzyme Q. Okay, when it gets its electrons, it looks like a Pac-Man character, right? So there we go, one more, and there you go. There's your Pac-Man character. You see what I'm saying? But that would be fully reduced coenzyme Q. Okay, and so it's just going to keep receiving these electrons from this one at a time, and then it's going to go take them off to complex 3. Okay? Oops. All right, and that's it. That's all I need to say about that. Okay? I'll try and remember to post that on Canvas. If you don't see it like at the end of today or by tomorrow, write me an email because I forgot. Okay? But I'll try and remember. And then maybe David Letterman in mathematics will show up too in your account. Okay. Questions on that? Now, you'll notice in that depiction there, coenzyme Q, it's important that it can also adopt the free radical form because it has to accept electrons one at a time. Okay, so the utilization of these iron sulfur centers dictates that only one electron can be moved at a time. And so things that are going to carry 2 electrons have to be able to accept them one at a time. Or donate them one at a time. Okay. All right. And that's it for complex 1. Okay. Awesome. Questions? Nope. Okay, let's do complex 2. Complex 2, we're going to move through rapidly. Okay, so because complex 2, in terms of mechanism, and how electrons get through the complex is really very, very similar to complex 1. It's actually less complex than complex 1. Okay, so let's look, though, first of all, at what the complex is going to do. Succinate, okay, is going to bring the electrons to complex 2. Now, remember, complex 2 is the enzyme succinate dehydrogenase. This is the citric acid cycle, step 6. Okay. This is the only citric acid cycle enzyme that is not a soluble enzyme found in the matrix of the mitochondria. It's embedded in the inner membrane of the mitochondria. So we always draw the citric acid cycle as this nice little ring, right, or this circle. In reality, that circle should be drawn up to about step 6, and then it should have this loop that goes all the way to the electron transport chain and then come back. Okay. Because that's indeed what's happening. Succinate goes to the membrane. Okay. Succinate will then be oxidized to fumarate. Okay. And those electrons that enter into complex 2, that enter into complex 2, okay, will reduce coenzyme Q. So this will be again reduced. And succinate will be oxidized. Okay. Now we always draw in complex 2, we always draw FADH2 as the product of that. Okay, but it's actually an intermediate step in this. Okay. The product is QH2. Okay. Now the enzyme itself is actually pretty small relative to complex 1. So it is comprised of 4 subunits. There are 3 iron sulfur centers, and then there is 1 FAD, and then finally 1 heme B. Okay. We'll look at the structure on our next slide. Now, I'm not going to draw arrows and all that stuff and do all that stuff. I'm just going to say this because it would be really very, very similar to what we've already done. Here's what I will say, though. Okay, we'll start at succinate. And succinate will give electrons to FAD. Okay, when it does that, it will become fumarate. Okay, fumarate will leave then the enzyme active site. Electrons will come to FAD. That FAD, once it gets its electrons, will become FADH2. Okay? The protons come from the succinate. Okay? it will give up its electrons one at a time to a chain of iron sulfur centers. Okay? And there's 3 of them, so I'll just put 3. And those iron sulfur centers will again transition between iron 3+ oxidized form to iron 2+ reduced form. Okay. Same jumping mechanism though, right, that we saw in complex 1 that hopefully you have a visual for now that we've gone through a video and of course our analysis on complex 1. Okay. Finally, the last thing that will happen here is that those electrons, one at a time, will reduce coenzyme Q, making QH2. Okay? This will go on to complex 3. Okay. It needs to get 2 protons from the inside to do this. That's a very quick description of how electrons make it through this. Okay. One of the key points here that needs to be made is that there are no protons pumped. Okay. Meaning translocation of protons from N to P side does not occur at complex 2. It's the only complex that it doesn't do that. Okay. There's 4 in complex 1, as we just saw. We'll look at complex 3, there's 4 there. And there's 2 by complex 4, but there's none by this. Okay, there's the only one. Questions? Structurally, we could look at this. This looks really similar, okay, to what we looked at with complex 1. I mean, in some regards, it looks very similar. We have here iron sulfur centers. We'd have here, this guy would be FAD. And then finally, it is shown here, coenzyme Q. Okay, so electrons again come in as succinate. Give the electrons to FAD. Fumarate leaves. The electrons jump up this until finally they get to Q. This becomes QH2. And off it goes to complex 3. Okay. And that video will show the mechanism of this as well. Okay. Now, real quickly, okay, that's the mechanism. I do want to show this. I'm going to highlight this here. I'm going to mention it also for complex 1. This is heme B. And if you'll notice in the last slide and in this slide, I have not mentioned that heme B receives or donates electrons. It doesn't. It's not on the chain of electrons. You also hopefully remember that in complex 1, I said 8 of the 9 iron sulfur centers are used in the transfer of electrons, but there's another one, there's one that isn't. Okay. So the question regarding, for instance, heme B and that extra iron sulfur center is, well, why are they there? And evidence seems to point to the fact that these are antioxidants. They are, in effect, electron mops. Okay? We're going to talk about, certainly not today, but more than likely Friday, we'll talk about oxidative stress and the fact that the electron transport chain is a generator of oxidative stress. And oxidative stress can be, you may know this, very harmful. Okay? And so there are a number of mechanisms in a cell that are designed to reduce oxidative stress. One of the proposed theories regarding this additional heme B or that additional iron sulfur center is that these reduce oxidative stress. In fact, with complex 2, I can't say this for complex 1, but with complex 2, there is genetic mutation where that heme B is not found in that complex. Okay. those individuals that have that genetic mutation suffer from tumors. The development of tumors at younger ages, younger than you would expect. Not always cancerous, but they can be benign, but they're still large growths, specifically, strangely, in the neck and the head area. And I'm not sure why it would be more prevalent there, but again, the proposal there is that, well, you lack this antioxidant in complex 2. There is increased oxidative stress that leads to increased mutation, that leads to then uncontrollable growth, cellular growth, tumors. Okay. Questions? No? Okay. The Rockies were in the 10th inning when we started today. I just want you to know that. So they haven't had a very good 4 days. I want to be honest with you. Okay, they've lost 4 games in a row. It's been pretty bad. I think that they need to have someone explain to them that to win a baseball game, you have to score more runs than the other team. So, oh, did they? Oh, okay. Well, that's good. That means they scored more runs than the other team. So I'm thinking about, I think I could be like a consultant, professor baseball or something like that, you know, and just to tell them those kind of things. You know, there's 2 main strategies. We either have to prevent the other team from hitting the ball as much as we do, or we have to hit the ball more than they do. And that sounds like the same thing, but it's not actually. Okay, so, but I think that I could get hired to be professor of baseball. I would ask for less money than Nolan Arenado. I mean, not significantly less, but less. Okay, this is a summer gig for me, though. I'll just do it in the summer. Yeah, question. Okay, so the question is, wouldn't oxidative stress just kill the cell at high enough levels? Yes, and we'll talk about that. But you can have lower levels of oxidative stress that will actually can just lead to errors, more significant errors, in specifically genomic information that can then lead to abnormalities in terms of cellular growth. Okay, depends on the level. It doesn't take very much oxidative stress to kill it, though. Okay? But it can lead to mutation that leads to cancer or benign tumors in this case. That was very good of you to bring us back away from baseball. So, no, what's that? No, it's good. Someone's got to rein me in sometimes. Anyway, Professor Baseball. On my CV, I'm #1 in my fantasy team right now, just so you know. I mean, that's why I feel that I can tell people what to do. You know, fantasy baseball and real baseball are about the same thing. Okay, anyway. Questions? No. Okay, great. Well, then let's move on. Okay, let me ask you a question, actually. Okay, I have a question for today. Yeah, okay, this is it. All right, save changes, sure, why not? Okay, let me pause this. So go back into your notes. So I want you to go back to the glycerol 3-phosphate shuttle. And you think, wasn't that before spring break? Exactly. Sometime before spring break, go back to that and tell me what the final electron acceptor for that process is. Okay, so go look at that. Text me your answer. I'll give you 1:40 seconds to do that. On your marks, get set, go. All right, you got about 20 seconds left, so submit an answer. Even if you haven't really found the answer that you want, submit an answer so you get credit for being here today. 10 seconds left. Okay. 3 seconds, 2 seconds, 1 second. 74. Okay, well, let's look at this. Let's check it out and see what people put. Okay, a lot of you saying Q, coenzyme Q, FADH2, QH2, oxygen. Very good. I don't remember. Right, Q. Nolan Arenado is not no. Actually, I did verify that the Rockies won 1-0 in the 11th inning. Good job, Rockies. Okay, back on the winning track. Okay, anyway, ubiquinone. Lots of you are saying ubiquinone, Q. Okay, so in the interest of time, let's just cut right to the answer. Q is indeed the answer. Okay, or Q going to QH2. So if you look back at those notes, the end results of electrons are going to be transferred from, again, NADH to go through the glycerol 3-phosphate. The glycerol 3-phosphate gives electrons to FAD, making FADH2. FADH2 gives electrons to Q, making QH2. Now, those electrons, I think the answer right here, this is actually right. I mean, those electrons will go on through complex 3, complex 4, and eventually make it to oxygen. Okay, so they're entering, though, the electron transport chain. Now, I bring that up, and the reason I bring that up is because we have just seen this. Okay, this is a figure here. We just have seen how complex 1 leads to the production of QH2. So we could call this QH2 if you want. Coenzyme Q works too. Okay, we just saw also how complex 2 can do that. Okay, well, it turns out that cytosolic oxidation, and this would be glycerol. I'm sorry, not glycerol, but this would be a glycolysis leading to the production of NADH. It can enter into this glycerol 3-phosphate shuttle. Those electrons can make it to glycerol 3-phosphate dehydrogenase, to FAD, to then QH2. Okay, and then finally, and there's one thing I should just, this is not right. Okay, this figure's wrong. Okay, so that's not right, but this is right. Okay. And that is that fatty acid oxidation, we'll see this, what we call beta oxidation, it'll pass electrons to something called ETF, electron transferring flavoprotein. Electron transferring flavoprotein will pass electrons to electron transferring flavoprotein ubiquinone oxidoreductase, which is fun to say and spell. Don't worry about it, it's written down there, ETF, don't worry about the name of it, but, okay, those will pass electrons on to coenzyme Q, making ubiquinol. Now, the whole point of this is that electrons from many different oxidative processes in the cell will all funnel into this molecule, ubiquinol. Okay. Now, it did say amino acid oxidation. It's not right to say that it's going to pass electrons to ETF. Amino acids are going to feed into either the glycolytic pathway or the citric acid cycle, and those will feed into either complex 1 or complex 2 and make it to ubiquinol as well. So you can fairly say that pretty much every oxidative process in the cell that's going to lead to oxidative phosphorylation, all of those electrons will be funneled into ubiquinol. Okay. From that, then they'll start being passed to complex 3. Okay. Right, so that's this funneling down process. Okay. All right. Questions on that? No. All right. Let's start complex 3. We're certainly not going to finish complex 3. Okay. We'll get a little bit of a ways into this. We'll definitely finish it on Friday and complex 4 and talk about oxidative stress as well. Okay. So complex 3. The name of complex 3 can also go by cytochrome BC1. That would be a name that you might find in primary journal articles or maybe even in other textbooks. We'll call it complex 3 here. It's going to, of course, take that ubiquinol derived from the processes that we just talked about. It's going to oxidize it. Okay? And it's going to pass the electrons onto 2 moles of something called cytochrome C. So this is going to be reduced. Okay. This cytochrome C is a peripheral membrane protein. Really small, actually. Okay. When it receives these electrons from ubiquinol, and you have to have two of them because there's cytochrome C has a heme group in it, heme C, and it can only accept one electron at a time. You may also recognize, if you've covered apoptotic pathways, cytochrome C is an important aspect of apoptosis. Okay, so same molecule. Okay, we're not going to cover apoptosis in this class. We're not really cell biology in that sense, but we are looking at that same protein and its role, functional role in the electron transport chain. In totality, there are also 4 protons, all of which are going to end up on the P side. Okay. Now, the complex. Let's go ahead and talk a little bit about the enzyme itself. It's 22 total subunits. So it's pretty big. Okay. It's often thought of, though, as a dimer of 2 11 subunit functional units. Okay, so it's a dimer of 11 subunits, of 11 subunit complexes. Okay, meaning you have 11 subunits, and then there's another 11 subunits. They all come together, but there's 2 copies of each subunit. Okay, and so it's really symmetrical molecule that you can almost virtually cut in half in terms of what it would look like. So 2 copies of each subunit. Okay. Now, subunits. Okay, there is a cytochrome B subunit. 2 of them. Okay. And in each of those subunits, there are 2 heme groups. Heme, what we call BL, and heme BH. Okay. There is a cytochrome C1 subunit. Okay, that has a heme C1 in it. And then finally, there's an iron sulfur protein. And in the iron sulfur protein, there's a 2 iron, 2 sulfur center. Okay. It's fairly complicated. Okay. I want to make sure that we understand one thing. Cytochrome C1, okay, is not the same as cytochrome C, despite the very similar nomenclature. Okay, that subscript 1 is very important. Okay, so let's end there. That's a perfect place to go ahead and stop, and we'll come back and we'll look at something called the Q cycle. Remember, quiz 9 is not due till Friday. So probably wait on that. We haven't quite covered all the material for that.